All right. Hey guys, welcome to chapter 3, the cell cycle. So chapter 3 contains 4 parts. 3.1 looks at an introduction to why cells divide. Chapter 3.2 looks at the cell cycles and the specifics of those processes. Chapter 3.3 looks at controlling cell division. And then 3.4 rounds it off with looking at stem cells. So let's jump into it. All right, 3.1 cell replication. Here is a picture showing the different stages of human development, and as you can see, that kind of cycles through 23 different stages leading up into 56 to about 60 days of the development of the embryo, starting from day 1, the zygote that's here, and then it kind of works its way through. Pretty fascinating photo, which I just want you guys to have a look at and just kind of notice and see how the human embryo develops over the course of these stages. And as you guys look at that, I just want you to ponder this particular question. When does life start? All right, when does life start? At the formation of the joining of the sperm and the egg, or perhaps maybe at birth or at some other development stage? And it's just a question for you guys to wonder and to think about as we progress through this particular chapter. Okay, so let's take a little bit of a look at cell replication. You can see in that little animation there how over time the development of a newt occurs in the egg, right? Back in chapter 2, we looked a lot at cell theory, and one of the things we actually looked at was the idea that all cells come from pre-existing cells, and we call that biogenesis. And that's exactly what we're going to zoom into in this particular chapter. So cell replication is a form of cell division in which a parent cell divides and is going to produce genetically identical daughter cells, right? And there are kind of 3 main reasons why cells choose to replicate and divide. The first one is the nucleus to cytoplasm ratio. The second one is growth and development. And then the third one is to maintain and to repair damaged or lost existing cells, right? Now, in unicellular organisms, cell replication is also a means of reproduction. In other words, that's actually how they produce offspring. But in multicellular organisms like us, for example, cell replication is it encompasses those 3 particular reasons. And then there's an individual or a separate process for reproduction, which we will look at in the later chapters, okay? But here's a little sequence to show what happens in day 1, the start of day 2, and you can see 1 becomes 2, 2 becomes 4, 4 becomes 8, and then eventually you get a very, very kind of large complex of cells starting to form what looks like an embryo, okay? So let's have a look at these particular reasons, and that's the focus of this particular section, okay? So the first reason for cell replication is actually the nucleus to cytoplasm ratio, right? As a cell grows larger, the surface area to volume ratio decreases, and as well as the size of the nucleus relative to the cytoplasm. In other words, as the cell gets bigger, the cell itself and all of its contents gets bigger, but the nucleus doesn't actually increase that much in size. So that means there's a few problems that the cell actually begins to encounter. The first is that the synthesis of proteins and nucleic acid doesn't keep up with the size of the demands of the cell. So it demands more protein and things to grow, but it can't produce as many because of the difference in the ratio between the nucleus and the cytoplasm, right? It also means that as it grows bigger, the distance for transport, the movement of nutrients kind of in and around the cell, increases. Just like how a city gets bigger, it becomes harder to move resources around the city. When a cell gets bigger, it becomes hard to transport nutrients and other materials needed for the cell without taking up more time. And so that exchange of material becomes inefficient, right? So cell replication allows the nucleus to cytoplasm ratio to actually be restored, right? So as it grows, becomes more inefficient, halves, and then that ratio becomes restored, and then it continues to function efficiently. So every time a cell kind of gets up to a certain size, it would want to replicate rather than continue to grow. Now, after fertilization occurs, the first new cell of an organism, that's actually called the zygote. And as it contains all of the necessary nutrients of the cell, that first zygote, that's actually a really big cell. For an average cell, it starts off as being really big. So when you see the zygote beginning to replicate and divide, it actually starts to grow smaller until it reaches this kind of stable point for a cell size, right? So here is a diagram showing you that process of when it first fuses together. And you can see there's your sperm, and then there's your egg, right? The egg contains all of the other stuff, and the sperm just pretty much has the genetics. And then around about here, you can start to see it starts to divide. And then here, you get your first division, and then 2 becomes 4, and then 4 probably becomes 8 somewhere around here. You can't see it because they were hidden behind the ones that are there. And then eventually you get to a stage where you start to see the development of the embryo separate from the development of the placenta, right? And those particular stages are now considered as when the cell begins to specialize, right? Now, second reason for cell replication is just simply growth and development, right? When multicellular organisms grow and increase in size, they also must increase in the number of cells that they contain, right? Because of that same reason, the nucleus to cytoplasm ratio, it doesn't make sense for you, the individual, to be made up of the same 15 cells or 16 cells you started off with that are just oversized cells. So rather than that, they multiply into several thousand, hundreds of thousands of cells to form the different parts of your body. The third reason is actually maintenance and repair. So inevitably, cells are going to die off because they either reach the end of their lifespan, or they become damaged, or they become infected, and therefore new cells need to be replicated to replace the old ones, right? The rate of the replication is going to vary depending on the type of tissue, and the extent of repair also varies between organisms, right? So, for example, right, your epidermal cells, your skin cells, you have a dermal layer below that, and the top layer is actually dead, and they actually flake off all the time. You lose lots and lots of skin cells every single day. And so the ones below that, the dermal layer, the one that's growing, needs to then replicate to replace those dead skin cells and kind of shuffle their way towards the surface of your skin. And that's actually what your skin kind of looks like at the surface where it's dead. And look, there's a bacteria right there, right? The third one is a little gif of a starfish, and you see this starfish, that big giant arm, that's the original arm, and you can see the starfish has actually lost its other arms and has actually regrown those other arms. And so long as a central part of the central disc remains, the starfish actually can continue to regrow its arms and things like that, right? The table on this side here, you can see the average lifespan of a few of your different cell types, right? So for example, your intestinal lining, your intestines, the cells that line them to, absorb nutrients and so forth, they only last about 1.3 days. So in other words, whenever the food that you eat kind of moves its way through the intestines, it inevitably rubs off and removes a few of them as it goes. And then also, stomach lining lasts about 3 days. Your tongue, 3.5. You notice where all the abrasive surfaces tend to lose their cells quicker, whereas a few of your other ones last a little bit longer. For example, your red blood cells, they last about 120 days. Yeah, that's about 120 days, about 4 months, right, before they reach the end of their lifespan and need to be removed. Whereas other cells, for example, your nerve cells in the brain, they last your entire life. Your lifespan, right? Makes sense, because if you were to lose nerve cells in your brain, you would probably lose things like memory, and that's probably not a great thing. So you want to be able to not, replicate them too frequently, otherwise you would forget things like no tomorrow. Now, for prokaryotes, a bit of a sidestep, the process of cell replication actually has another name. It is called binary fission. Binary meaning 2, fission is to divide. If you were to fissure something, you'd break it into 2. So prokaryotes, like bacteria and archaea, are simple organisms, and they're single celled, which means the replication is going to be much quicker and far more rapid than your eukaryotic, more complex counterparts, right? And so what it kind of looks like is that the prokaryote contains one strand of circular DNA, which is called a plasmid, and it is found in the nucleoid region, and that's going to be attached to the plasma membrane via the origin. So there's the attachment site that's there's the nucleoid, and then what happens is the DNA molecule here is going to be replicated first, giving you 2 identical DNA molecules within the one bacterial cell, and then the cell is then going to elongate kind of stretching itself out, and the DNA molecules are going to be pulled to the opposite sides of the cell, and then a new cell wall is going to form in between the two in a sort of pinching fashion, and then eventually it's going to split apart, forming your two daughter cells. Now, because that replicated DNA is essentially identical to one another, that means the two daughter cells are going to be exactly the same as the parent cell. Now, there are mutations that occur between the two, so there are going to be slight changes between the generations, but in general, you're going to get identical daughter cells from the original parent cell. And this process, that takes about 20 minutes. So if you have one bacteria, within 20 minutes, that's going to become 2, and after 40 minutes, it's going to become 4, and then by the end of the day, you're going to have about 10,000 bacteria sitting on your little agar plate or your little surface on the table or something like that, right? So it's a pretty rapid process. Now, the eukaryotic process of cell replication is far more complex, and that's actually what we're going to cover in the next topic in 3.2. So I will catch you guys then, and enjoy the rest of your day.